Assalamualaikum warahmatullahi wabarakatuh. My name is Tuan Nurhayati binti Tuan Mahmud. I would like to present my final year project entitled Evaluation of PCR Amplification based on primer SSR-SMG00042 through direct PCR and indirect PCR method. Oil palm agriculture is expanding rapidly across the world. With many unsustainable oil palm planting activities that existed today, it is necessary to produce oil palm plants with improved quality. High-quality DNA is the key for successful molecular experiments. The traditional method to handle experiments, for example, preparation of chemicals and buffers in experiments and sample collecting activity is challenging, includes time-consuming, cost-consuming, and storage handling issues. To solve this issue, many alternatives have been developed. Existence of extraction kits and FTA cards give huge distribution to plant improvement research. Emergence of more advanced DNA technology caused the technological revolution. This is the good sign. Breeding program that has been developed helps palm breeders to develop improved cultivars of plants and a variety of oil palm that has been commercialized for economic use. However, the existence of many plant cultivars make it difficult for humans to distinguish oil palm varieties phenotypically. Problems arise when the independent smallholder tend to be scammed during seedling purchase. Poor seedlings will cause low productivity. It will cause lands become more limited. To solve this problem, scientists study polymorphism studies. However, molecular DNA experiments need to be done correctly. Isolated DNA extracted from plant samples is not easy as plant cells have cell walls, making them hard to break. Despite of DNA isolation and PCR being developed a long time ago, protocols to get high-quality DNA that are easier as well as best PCR conditions are still improved by researchers until today. The objective of this study are first, to evaluate PCR product based on primer SSR-SMG00042 through direct PCR and indirect PCR methods as well as determine annealing temperature of primer use. Second is determine the suitable voltage to be used in electrophoresis 2% agarose gel loaded with oil palm samples. In this study, 3 PCR sessions were done. First method labeled as PCR1 is DNA isolation and amplification based on primer SSR-SMG00042 using direct PCR method with the use of FTA cards. Second method labeled PCR2 is the DNA isolation and amplification based on same primer using direct PCR method with the use of TransDirect plant tissue PCR kit. Third method labeled as PCR3 is DNA isolation and amplification based on the same primer using indirect PCR method with the use of PlantZol kit. In method 1, oil palm leaves were crushed onto the FTA cards and being processed. Meanwhile, in the method 2, frozen oil palm leaves are directly used to process. In method 3, fresh leaf processed and continue with DNA quantification. After that, all samples undergo PCR, proceeding with electrophoresis in 2% agarose gel, and then visualization of DNA bands using an imaging system. Primer used in this study was SSR-SMG00042 primer. This primer used because SSR markers are co-dominant. In polymorphism study, this primer's characteristics highly discriminative, make it able to distinguish polymorphism in close related members in population. Among these genetic markers in DNA, SSR markers has highest polymorphism information content, easy and faster to generate in PCR, easy to interpret its allele configuration, and transferably between laboratories. By using this marker, it would give some information related to polymorphism study of oil palm plants. Next, I will discuss the results obtained. Figure 1 shows the results of PCR1. From extreme left, 5 oil palm samples that has been amplified as well as 100 base pair DNA ladder has been run on electrophoresis gel. No DNA band obtained by sample 1, 4, and 5. DNA bands obtained in results from sample 2 and 3. Bands obtained from 2 out of 5 samples show that the FTA mini card used done a good job in collecting and preserving the plant sample due to its technology. However, non-efficient PCR results as well as non-efficient DNA isolation has been obtained by samples through this direct PCR method using FTA cards. Problems have been identified. Solutions and reagents used during isolation and washing step are contaminated, thus influencing enzymatic reaction during DNA amplification. Figure 2 shows the results of PCR2. From extreme left, 100 base pair DNA marker loaded into well 1, continued with other 5 oil palm samples that has been amplified, been run on electrophoresis gel. No DNA bands obtained in samples based on kit supplier transgene, the kit suitable for plants with moderate content of polysaccharides. DNA might not be isolated well as oil palm tissue contains high contents of polysaccharide. Table 1 shows DNA concentration and absorbance at 260/280. Absorbance value should be between 1.7 to 2.0 to be considered as pure DNA. Step continue with PCR despite having unfavorable DNA purity and DNA concentration value because of time limitation. Figure 3 shows the results of PCR3. From extreme left, 100 base pair DNA marker loaded into well 1. Continue with other 8 oil pump samples. Sample 1, sample 2, sample 5 and sample 6 shows successful PCR. However, sample 3, sample 4, sample 7 and sample 8 shows failed PCR. One of the reasons is inefficient DNA isolation. 4 out of 8 samples obtained DNA bands. PCR bands obtained using this PlantZol kit method were better. The forces applied during grinding step as well as disruption of cell wall by liquid nitrogen has released more DNA. After first gel being visualized, voltage applied for the next step electrophoresis being adjusted to respond to effects that has been produced. In figure 4, suitable voltage for electrophoresis evaluated by observing the DNA bands obtained on the oil gel. Figure 4a and figure 4b, 9 microliter and 7 microliter DNA ladder were loaded respectively. Results obtained were overlap and blurry DNA marker bands. In figure 4c, voltage reduced to 70 volt, volume 100 base pair DNA ladder reduced to 1 microliter. DNA ladders when obtained are smaller, non-overlap and clearer. After electric field being applied, DNA traveled from negative electrode toward positive electrode. Voltage increased, migration rate on gel increased. Voltage rates are too high, causing problems. From the observation, when voltage reduced from 90 volt to 70 volt, suitable voltage applied has given better effects on the DNA marker band visualization. As a conclusion, direct PCR method using FDA card and indirect PCR method using PlantZol extraction kit are recommended method to be used in processing DNA of oil pump plant for amplification purpose. Second, annealing temperature for SSR-SMG00042 primer was not obtained specifically, but it lied between 49.6 degrees Celsius to 51.8 degrees Celsius. In addition, to run 2% 250 ml gel during electrophoresis, approximately 70 volt voltage is suitable voltage. Meanwhile, volume of the DNA ladder should be more than 1 microliter but less than 7 microliter. As recommendation, fresh sample should be used to yield more DNA. Most importantly, repeat gradient PCR because it is the best option to find the perfect annealing temperature for amplification of primer.